All right, now it's my pleasure to introduce Dr. Tina Mueller. As a scientific advisor, she consults with customers and internal teams on in vitro ADME programs with a focus on drug-drug interaction studies and compliance with regulatory guidance. Before joining BioIVT, Dr. Mueller was an associate director of research at Cybin, where she led the company's research program from early through late discovery. Additionally, Dr. Mueller served as a research associate professor at Michigan State University, senior staff scientist at Scripps, Florida, and a professor of biomedical sciences at Kaiser University. Dr. Mueller obtained her PhD in biochemistry and microbiology and earned a master's degree in environmental sciences from the Swiss Federal Institute of Technology in Zurich. Tina, I'm looking forward to your presentation.
Yeah, thank you so much for the introduction, Michael. And hi, everyone, and thank you for joining. As Michael just said, I am Tina Müller, and today I'll be talking about protein degraders in drug discovery, focusing on their mechanisms, ADME challenges, and translational strategies. I hope you will enjoy this webinar, and I'm looking forward to your questions at the end. So let me start with a quick overview of today's presentation. I will first briefly introduce protein degraders as a therapeutic modality, and then focus on key ADME considerations specific to this class of molecules. Next, I'll walk through a case study highlighting how we adapted ADME workflows using two PROTACs, ARV-825 and JNJ-1013. And finally, I'll summarize the key findings and takeaways. So let's get started. So in drug discovery, we traditionally relied on small molecules to inhibit protein function. But as we probably most of us know, this strategy only works when a protein has a suitable binding site, and many simply don't. So in fact, nearly 80% of disease-relevant proteins are considered undruggable, including transcription factors and scaffolding proteins. Now, protein degraders offer a fundamentally different solution, as they, instead of inhibiting the protein, they eliminate it entirely, and they do so by harnessing the cell's ubiquitin proteasome system. That also means that they expand the accessible target space into what was previously considered undruggable. Now, as a biochemist, I find protein degraders fascinating for especially the reasons I just mentioned. But until very recently, the modality was, if you want to say so, just a concept for drugs in development. Now, just a little more than a month ago, though, that has changed, and an important milestone was reached: the first FDA approval of a heterobifunctional degrader, or PROTAC. Now, this represents the first regulatory validation that heterobifunctional degraders can deliver a clinical benefit, and it also demonstrates the successful translation of a protein degrader from preclinical development to clinical therapeutic application. And to better understand how we got here, let me walk you through some key milestones. If we go all the way back to the 1950s, thalidomide was developed and used to treat morning sickness that unfortunately caused severe birth defects. Now, with that said, at that time, thalidomide's mechanism of action was unclear, and it was considered a small molecule drug. Indeed, it took quite a while to elucidate its different mode of action and the fact that it indeed eliminates its target and not just simply inhibits it. Now let's fast forward to 2001, when we got the first proof of concept study for targeted protein degradation from Sakamoto, Cruz, and Desai. They described peptidic PROTACs, where one end binds the target protein and the other end was a phosphopeptide, which was recognized by an E3 ligase. However, these first generation PROTACs had significant limitations due to the peptide-based ligand. Specifically, they had poor cell permeability and limited stability. Now, in 2004, the field expanded to other ligases with the first VHL-based PROTAC. but still it was using a short peptide to recruit the ligase. So while conceptually important, the molecule was still not yet drug-like. Now that changed when the turning point came around 2008, when we started to see fully small molecule-derived PROTACs. So in other words, both ligands were small molecules. And this was really the moment when PROTACs became more drug-like and tunable. And from there, the field accelerated quickly, and companies like Arvinas played a major role in pushing degraders into the clinic. So between 2019 and 21, the first PROTACs entered clinical trials that included ARV-110 and 471. And in 2020, ARV-110 progressed into phase 2 clinical development. And 2 years later, vepdegestrant, which is ARV-471, entered late-stage clinical studies. And then more recently, just last year, the NDA application for vepdegestrant was accepted. And as I mentioned before, on May 1st, the FDA approved the drug. So in other words, the first PROTAC progressed all the way through from development to approval. And equally important, I think we are just at the beginning of what protein degradation can achieve. So before we go into the science and the case study, I think it's worth briefly looking at the development landscape. Targeted protein degraders are no longer a niche concept that is dominated by academia and small biotech. As you can see here, large pharma is making substantial investments in the space. And what I find also particularly interesting is the breadth of these investments. They are not limited to one therapeutic area or one type of degrader. We see activity across heterobifunctional degraders, molecular glues, and even newer concepts such as degrader antibody conjugates. Now, while the examples here are not meant to be exhaustive, but they do illustrate the level of attention the field is receiving. Companies such as Novartis, Roche, AbbVie, Lilly, and Amgen are making sizable deals through collaborations, licensing agreements, as well as acquisitions. So many companies, including large pharma, are very much interested in degraders as a modality. And I find this especially noteworthy because until just a month ago, no approved drug was actually on the market. Now, talking about approval, let's transition a little bit and look at where the field is clinically. Here we have a high-level overview of the current clinical landscape with a couple of key points that characterize the space. First, the field is growing quickly, but when we look a little bit closer, clinical trials are still predominantly in early stages. So that means most programs remain in phase 1 or phase 1-2, and only a small number actually have reached late-stage development. So while the modality is overall advancing rapidly, we should consider the field as clinically still emerging. The second point is that oncology is the dominant indication, and most early degrader programs were built around well-validated oncology targets. Examples here are ER-positive, HER2-negative breast cancers, castration-resistant prostate cancer, as well as hematological malignancies. Now, with that said, and I'm happy to see that, the field is now moving into immunology and inflammatory diseases. A third point I would like to raise here is target concentration, and that's somehow related to the second point. But the clinical space is really focused around a relatively small number of validated targets. Now, this includes the estrogen and androgen receptor, BTK, as well as Icaros. Now, in other words, the field is building proof of concept molecules, and hopefully that will build confidence in the modality to expand, hopefully soon. And then last, there's also limited diversity in the E3 ligases, with just two which are regularly used, cereblon and VHL. Now that has practical implications for selectivity, tissue distribution, and also possible resistance building. So now let me introduce heterobifunctional protein degraders, also called PROTACs. And just to state it once clearly, I will use the terms interchangeably. And as you probably also figured out by now, we will focus on this type of protein degraders for the remainder of today's talk. So here on the left top, there is a simple figure illustrating the structure of a PROTAC. The molecule consists of two ligands, which are connected by a linker. Now, one of the ligand binds to the protein of interest, or POI, which is shown here in blue. You can see that here. And the other binds to the E3 ligase, which is shown here in yellow. Now, in the middle, we have the structure of ARV-110, which is a typical PROTAC molecule with the different parts shaded as above in the simple structure. Then last, let me point out some of the physicochemical differences to typical small molecules. PROTACs are generally considered beyond the rule of 5 molecules. So in other words, they have a high molecular weight, higher polar surface area, and more rotatable bonds. Then on the right-hand side here is a key feature of protein degraders, which I would like to point out up front here, which is the so-called ternary complex, where the protein degrader is bound to the protein of interest, shown here, and the E3 ligase, shown here in purple, at the same time. Now that we have seen what PROTACs look like, the key question is, how does this design translate into the pharmacology, and how does it differ from traditional small molecules? So protein degraders operate through an event-driven mechanism, which brings the protein of interest into proximity with the E3 ligase. Now that will lead to ubiquitination and elimination of the target protein, and more on that on the next slide. What I would like to emphasize here is that this mode of action shifts the pharmacology from occupancy to event-driven pharmacology. As a result, there's a catalytic cycle, and only substoichiometric amounts of the drugs are needed. And the different pharmacology also expands the target space and allows enhanced selectivity. And again, I will explain that in more detail on the next slide. In terms of PK and PD, as well as safety, we often see nonlinear relationship with a prolonged pharmacodynamic effect that is dependent on protein turnover and resynthesis. Further, affinity alone does not necessarily predict degradation, efficiency, and just to mention that here as well, to make this list complete here, there's a potential for off-target effects as well as the risk of the so-called hook effect at high concentration. So building on what we just discussed, here is the mechanism of action that explains some of these implications in more details. Starting in the left top corner, the degrader simultaneously binds the protein of interest and the E3 ligase, forming the ternary complex, which you've already seen on 2 slides ago. Now, this illustrates the binding of the target protein alone will not drive its efficient removal, and that the critical parameter here is the formation of the ternary complex. Also, the possible additional selectivity of the molecule, as 2 events need to happen at once, and the orientation and binding to each is important. Last, I wanted to mention that for the target protein binding, no classical active site is required or needs to be accessible, as no inhibition needs to occur, and binding can be almost anywhere on the protein. Now, once the ternary complex has been formed, the proximity of the ligase enables the transfer of the ubiquitin molecules, shown here in green, onto the target protein, which leads to polyubiquitination. The ubiquitinated protein is then recognized by the 26S proteasome and degraded into small peptides. Now, importantly, the degrader is released during this process and can be recycled to engage additional target molecules or target proteins, which results in the catalytic event-driven mechanism of action and also substoichiometric degradation. So how do we approach in vitro ADME evaluations for these molecules? The overall strategy is to evaluate analogous to small molecules. That means assessing metabolism and drug-drug interaction risk by evaluating drug metabolizing enzymes such as CYP inhibition and induction, as well as transporters. And of course, supporting studies such as protein binding. However, assay conditions and timelines need to be adapted to account for the distinct properties of these molecules. So early in development, there are challenges with solubility, stability, and non-specific binding that need to be assessed ideally in preliminary evaluations. Cell permeability is also often challenging as it is often limited and critical to evaluate early in the discovery or in the development, as these molecules are often developed as oral drugs. Now, with that said, and complicating the situation here and the evaluation, traditional small molecule assays such as KCO2 tend to be poor predictors for in vivo behavior. Next, we have high protein binding is also common and should be evaluated early. And finally, complex metabolic pathways make both stability assessment and metabolite identification more challenging, which requires tailored strategies and, of course, excellent bioanalytical expertise. So with that, I'm going to present the case study that was conducted at our Kansas City facility. And before I start, I would like to acknowledge our team at that site who performed these studies. They are amazing, and I would like to give them a shout out before we go over the data. Now, the goal here is to show how we approached these challenging molecules and what hurdles we faced. I would love this to be the start of a conversation with all of you to get the best solutions on how to characterize these molecules most effectively. So what I'm going to do today is I'm going to present data obtained with two PROTACs, ARV-825 and JNJ-1013. And first, we address some of the challenges associated with the physicochemical characteristics, namely solubility, stability, and recovery. Then I will present data from our in vitro ADME studies, focusing on metabolism, metabolite profiling, CYP inhibition, and red blood cell partitioning. And at the end, I'll briefly go over assessing possible immune-mediated effects with data from a cytokine release assay. So let me introduce the two compounds that we used, ARV-825 and JNJ-1013, which both have the standard PROTAC design with two small molecule ligands and a linker. First, here on the left, we have ARV-825. The protein of interest is BRD4, and you can see that in purple, or the ligand in purple. The molecule has a little longer linker that is a PEG or polyethylene glycol in the middle. And then last, the molecule recruits cereblon with its thalidomide moiety, which is shown in the green box. As a side note, the thalidomide moiety has been described as susceptible to non-enzymatic hydrolysis in aqueous solutions, something to keep in mind. And last, some of the ADME characterization, in particular metabolic stability and metabolite profiling, has been done and published, so we can compare our results. The other test article was JNJ-1013. JNJ-1013 has a different POI ligand, which is shown here in orange, namely a moiety that binds ARV-125, and of interest, that molecule is considered to be highly cytotoxic. The linker is rigid, which is expected to make the molecule less cell permeable. And then it has a VHL recruiting ligand, which is shown on the right in green. So based on the expected challenges, we adapted some of our standard in vitro ADME workflows to better accommodate protein degraders. And the exact adaptations are here highlighted in teal. With that said, I included solubility assessment on this slide here to have a more complete picture. Even though that is routinely done for small molecules, I consider it important and wanted to include it here in the slide. In terms of the specific optimization, we evaluated the stop reagent during LC-MS development, method development. We assessed recovery from different test matrices, the stability of the PROTACs across different matrices, and added additional evaluation to the metabolism study with a no-cell control and separate analysis of lysate and media. So now let's go through the result. And up first, we have solubility assessment. The stock solutions were made in 80-20 methanol DMSO at a concentration of 2.5 millimolar. These were then tested at final concentrations of up to 25 micromolar in the incubation media, and all of that was done by visual inspection. And what we saw was that both compounds were soluble up to 2.5 micromolar. And you can see, just as an illustration, an example here on the right in that picture, where only the one tube on the very right would have passed that assessment and considered to be completely soluble. Next, we tested the stop reagents as part of the LC-MS method development to evaluate whether the compounds are stable after quenching. Our standard stop reagent is acetonitrile, so that's what we started with. And we also included acidified acetonitrile that contained 0.1% formic acid. Please note that this was done in one of the test matrices, namely Williams E, and the concentration of the PROTAC was 1 micromolar. And the final sample composition was 50-50 incubation medium to stop reagent. Now on the right, there are representative chromatograms with ARV-825 on the top and JNJ-1013 on the bottom. And both show the same sharp symmetrical peaks with and without acid, suggesting that acetonitrile is appropriate for use with these two compounds, and the addition of acid is not needed. So acetonitrile was used as a stop reagent for all experiments. Next, we tested all matrices to ensure that we have acceptable recovery of the two PROTACs. To evaluate that, we used two different setups. We had a test sample which mimicked the actual samples. So in other words, the PROTAC was added to the test matrix and then quenched with a stop reagent. Now this was then compared to the control sample for which the stop reagent was added to the test matrix first, and then the PROTAC was added. And the percent recovery was then calculated by dividing the test sample concentration by the control sample and multiplied by 100. We used two different concentrations for each matrix as appropriate for the respective study, which was in the range of 0.01 to 12.5 micromolar. And the matrices tested were CYP inhibition medium, Williams E, RPMI medium, and whole blood. And the results here are shown as graph bars on the right. On the top in blue is ARV-825 and JNJ-1013 is on the bottom, shown in green. The low and high concentrations are shown in the lighter and darker color, respectively. And what you can see here is that recovery was higher than 80% for each of the test matrices tested and also for both concentrations. In other words, no assay or compound-related issues with recovery was seen. Now to the next challenge, stability. We evaluated the stability in different test systems, and I show here 3, as some of it will be covered later. What I'm showing here is phosphate buffer containing magnesium chloride and EDTA, PBS, and whole blood. The PROTAC concentration was 1 micromolar. And as you can see again on the bar graphs with ARV-825 on the top and JNJ-1013 on the bottom, both of them are indeed unstable in phosphate buffer as well as PBS, with already considerable lower concentrations for phosphate buffer after even 4 hours and for PBS after just 60 minutes. In contrast, in blood, both PROTACs were stable over 60 minutes. Now, this highlights the importance of careful evaluation of this and selection of the test matrix when assessing the stability of protein degraders. So now let's move on to the actual in vitro ADME studies and talk about metabolic stability studies. We performed these studies in sandwich cultured human hepatocytes to capture both phase 1 and phase 2 metabolism and also to allow for longer incubation times compared to microsomes. Specifically, we used a mixed gender 10 donor pool. Now, based on published data, we expected rapid turnover for ARV-825 and therefore included early time points to capture that, and took samples at 0, 15, and 30 minutes, and then again at 1 hour, 2, 6, and 24 hours. We were also monitoring for cytotoxic effects, especially because the IRAK ligand of JNJ-1013 is known to have toxic effects. Now, based on cellular integrity throughout the 24 hours, no toxicity was observed. We also included a no-cell control, as 24 hours is quite a long incubation, and we wanted to ensure the stability of the compound and could confirm that both compounds were stable over the assay time of 24 hours. So now here are the actual metabolism stability curves for the 2 compounds. Again, on the top is ARV-825 and on the bottom JNJ-1013 in blue and green respectively. ARV-825 shows a single exponential decay with a half-life of approximately 1 hour. This is slightly longer than previously reported, and we attribute that to the different test systems, so suspension versus plated hepatocytes. Now, in contrast, JNJ-1013 follows a double exponential decay with an initial fast phase followed by a slower phase with half-lives of about 1.3 and 18.2 hours. Now, overall, the sandwich cultured hepatocytes were a robust and suitable test system to assess metabolic stability, and we were able to calculate half-lives for both compounds. Now, here are the results from the separate analysis of the lysate and media, which I mentioned previously. Starting with ARV-825 on the left, we see a rapid cellular uptake, which is shown here in dark blue here. It reached its highest intracellular concentration already at around 15 minutes and is accompanied by a rapid decline in the media as shown in the light blue points. Now, in contrast, JNJ-1013 shows slower cellular uptake, which is shown in dark green here, and consistent with its more rigid linker and expected lower permeability. The maximal intracellular concentration was observed later than for ARV-825, namely from about half an hour to 2 hours, before it gradually decreases. And we can see a concurrent rapid decline in the medium as shown in light green. So overall, the simple modification of a stability assay by separating the analysis of lysate and media provides useful information on cellular uptake, intracellular accumulation, and distribution. So next, I'll briefly outline the setup used for metabolite profiling. And just a disclaimer up front, I will keep this high level. I am not a mass spectrometry expert, but I'm happy to pass along any questions to our wonderful team. So we use the standard UPLC MS-MS workflow with a Waters Accuracy UPLC coupled to a CEVO G2 QTOF mass spectrometer. The chromatography was performed on a C18 CSH column under fairly typical conditions with a couple of tweaks. So we used an ammonium acetate-based mobile phase, but a rather standard gradient as shown in the table here. The mass spectrometry was then run in positive ion mode with a broad scan range to capture both parent compounds as well as metabolites. So overall, this is a fairly conventional analytical setup with a couple of important tweaks, so we could characterize the complex metabolite profiles of these molecules better. And here we have the metabolic scheme for ARV-825 with the parent compound on the top here in the middle. Now, overall, we identified 20 metabolites. 12 of these were previously reported, and we have shown them here in those blue numbers throughout the scheme. Then we observed 8 additional metabolites, and they are highlighted here in green. Now, on a high level, and not too unexpected, there are 2 primary pathways. We have O dealkylation and deamination. And as you can see, the major cleavage site in this molecule is the PEG linker, where the O dealkylation reactions are occurring, as we can see for many of the metabolites, such as C18 or C19, to just name a few. I would also then like to point out to the secondary and tertiary transformations. So the OH groups of the primary metabolites, they are further metabolized to carboxylic acids. So for instance, here from C18 to C13. And the carboxylic acid is then further oxygenated now on the actual ligand, so on the BRD4 ligand moiety. So for instance, C13 to C8. We also had one glucuronidated metabolite, which is from C17 to C10. And last, an amide hydrolysis metabolite down here in C9. When we switch to JNJ-1013, we see a slightly different metabolic profile. Overall, we identified 11 metabolites, and the main biotransformation included O and N dealkylations. We had amide hydrolysis and glucuronidation, resulting in predominantly primary metabolites. So we only see a few secondary and tertiary products, such as C4 and then C1 and 2. Also, in contrast to ARV-825, metabolism is primarily localized to the 2 ligand moieties, so with no significant cleavage observed in the linker. So compared to ARV-825, this highlights distinct metabolic liabilities between the 2 compounds and overall reflects the complexity of the metabolic pathways for these molecules. So now let's move on to CYP inhibition. For this study, we initially used our standard assay setup with a PROTAC concentration range of 0.025 micromolar to 25 micromolar, and we are using human liver microsomes. We had 3 different conditions, as is standard in our lab. We have direct inhibition which incubates the microsomes with the test article, the probe substrate, and NADPH. For time-dependent inhibition, or TDI, the microsomes and the compounds are pre-incubated for 30 minutes, followed by the addition of NADPH and the substrate. And for metabolism-dependent inhibition, or MDI, the microsomes, the PROTAC, and NADPH are pre-incubated before adding the substrate. So here we have the curves, and I'll explain them in a little bit more detail later. What I want to draw your attention here to, on this slide is what it looks like at the higher concentrations. When we look at the higher concentrations here, they appear, there is a plateau at that here, whereas the curves look okay at the lower concentrations. So in other words, the degree of inhibition does not increase between about 1 and 25 micromolar, which is not what you would expect. So this suggests that the assay conditions were not suitable, most likely due to solubility limitations of the compounds, and that we needed to make adjustments to the experimental setup. So after identifying the issue, we adjusted the concentration range to better reflect the solubility limits of these compounds and used 0.0025 to 2.5 micromolar. If we now look at the curves on the left, you can see a clear improvement in the data quality. For example, for CYP2C8, which is at the top, we now observe well-defined sigmoidal inhibition curves where inhibition increases consistently with the concentration, which is what you would expect for a typical IC50 experiment. And now for your understanding of these curves, on the left panel, we have direct inhibition, which is shown here in dark blue, and then time-dependent inhibition in green. On the right panel, we have again the TDI line as a comparator, and then metabolism-dependent, or MDI, in dark blue. Now, similarly for 2C19 and CYP3A4 and 5, the curves now show a proper concentration-response relationship, indicating that we are now operating within a concentration range where the compounds remain soluble and assay conditions are appropriate. And in the table summarizes the IC50 values, showing that for 2C8, 2C19, and CYP3A4, 5, we observe measurable inhibition in the micromolar range. Now, overall, adjusting the conditions allowed us to generate reliable IC50 curves, and it also highlights an important point. These molecules can act as precipitants, which needs to be carefully considered in their evaluation and risk for drug-drug interactions. Now, applying the same adjustments to JNJ-1013, we again see a clear improvement in the data. For CYP3A4 and 5, both with midazolam and testosterone, it resulted in nice IC50 curves. And I wanted to make you aware here that there is a left shift between the time-dependent and metabolism-dependent inhibition curves. The IC50 values are summarized. With that said, the IC50 values are summarized in the table on the right. And IC50 were also calculated for the metabolism-dependent inhibition by JNJ-1013, and they were clearly lower than the direct inhibition. So overall, similar to ARV-825, adjusting the assay conditions enabled generation of reliable IC50 curves, but again highlights the risk of the compound being a precipitant. And here we also have to include TDI as well as, or let's say, MDI. In addition, these data suggest that further follow-up, such as a reversibility or a Ki and K inact determination, may be warranted. Next, I just wanted briefly touching up on red blood cell partitioning. Given the complexity of PK-PD relationship for protein degraders, understanding their behavior in blood, including red blood cell partitioning, is essential. So we performed a standard red blood cell partitioning assay testing 1 micromolar of the test article at 0 and 60 minutes, and then evaluated the concentration in the red blood cell and the plasma. Looking at the bar graph, you can see the mean blood-plasma ratio at 0 and 60 minutes, and you can see that neither compound partitioned into the red blood cells. As controls, we had metoprolol and chloroquine, and they both showed the expected blood-plasma ratios. Now, let's switch gears for the last experimental data and look at some early efforts in the immune-related space. And we subjected the two PROTAC molecules to a cytokine release assay. One of the goals was that this can be an early de-risking study or evaluation specifically to assess the potential for unintended immune activation, which is important both in general and particularly for programs targeting I&I indications. In addition, we considered that this assay might actually serve as a functional readout for immune-relevant targets. So we used frozen human PBMCs and measured a panel of inflammatory cytokines across 3 individual donors, and LPS served as a positive control. The cytokine analysis was done with the Milliplex human high sensitivity T cell panel using the Luminex 100-200 system. And we evaluated the inflammatory cytokines, IFN gamma, IL-1 beta, IL-2, IL-6, TNF alpha, and IL-8. So here is one example of the response. We are looking here at the response for IFN gamma. You can see on the very left the solvent control in gray. Then ARV-825 is in blue, JNJ is in green, and the positive control on a different axis here, which has 2 segments, is shown in purple. And the different bars, so 1, 2, and 3, represent the individual donors. What you can see here is that both ARV-825 and JNJ-1013 showed minimal effects, with less than 2-fold changes, while LPS elicited a clear response, as you can see here, all the way to the top. And here are the other cytokines tested with the analogous bar graphs. And you can see that overall, neither of these 2 compounds activated cytokines, so showing no evidence of unintended immune conditions under these conditions. With that, I would like to briefly highlight where we are heading next and mention a couple of ongoing efforts in the lab. So here on the left, you see some of the ongoing studies which focus on addressing key gaps which we have identified. This includes expanding metabolic stability studies into whole blood. We also would like to evaluate cell permeability in MDCK2, MDR1 cells, and further optimize protein binding assays. On the right are some of the possible next steps, which are certainly of interest, and I would also love to have your point of view of what is most important in your mind. So one important area would be to expand the metabolism studies and look at cross-species comparison, which will be critical for the toxicology species selection. In addition, we could plan to expand into other ADME areas such as transporters and CYP induction studies. And of course, also we could evaluate a broader set of protein degraders. Finally, a key question for the field remains how predictive our current in vitro models are. So this is clearly beyond our in-house capabilities, but I would love to see some studies in that area. It would be so insightful to evaluate IV-IVE, particularly for or starting with clearance and metabolite profiling. So to wrap up, I'd like to highlight a few key messages from this work. First, early assessments for solubility, stability, and recovery, they were really critical to define appropriate assay conditions and also ensure the data quality. Second, relatively simple adaptations such as a modified hepatocyte assay and separating lysate and media analysis provided valuable insight into both metabolic stability and intracellular exposure. Third, we observe distinct metabolic profiles between the two compounds, emphasizing the importance of comprehensive metabolite identification or profiling and understanding compound-specific liabilities. We also had to optimize CYP inhibition assays to overcome the solubility-related artifacts and obtain reliable data for that DDI risk assessment. And finally, I think these results show that while protein degraders present unique challenges, standard small molecule ADME workflows can be successfully adapted with the right modifications. With that, I would like to acknowledge our fantastic operational team once more. First, we have our site lead, Joanna, who made the studies possible and provided great feedback on the results. Seema, Nadia, and Wei, together with Logan in the lab, are our excellent LC-MS experts who designed the bioanalytical protocols and analyzed the metabolite profile. Stephen served as a study director together with Melissa and made it all happen. And Leah was in the lab performing the assays, and Tyler performed the cytokine release assays.